So we'll start the committee. Nadine?
Thank you so much. Thank you to you both for excellent talks. My first question is to Dr. Martinez. You talked about GWAS and looking at gene sequencing in things like asthma, risk of disease. And one of the things that we understand is that the exposure to early adversity can, we're understanding one of the mechanisms can be through more epigenetic modification than actually things like, SNPs or things like that. And so my question is, are you familiar with any data looking at other ways of assessments of DNA accessibility, like ATAC sequencing or other things where we can actually look, given that this committee is looking at, the effects of environment and stress on the health and well-being of children, understanding the impacts of maybe the epigenetic modification in asthma symptomatology? Certainly.
I don't know if you remember, but in my winding road, the first of the three elements was epigenetics. And unfortunately, in 15 minutes, you cannot talk about everything. We're doing in asthma, many groups, but our group too, a lot of studies in determining if some of the early exposures that I was talking about, in a certain sense, exert their effects by permanently altering accessibility of DNA. And the results are very interesting. We've chosen the low-hanging fruit, in this case, maternal asthma, which is known to strongly affect, much more strongly in early life than paternal asthma, affect risk for asthma. And we have published results showing that mothers who have asthma transmit to their children changes in epigenetic markings in critical genes. But I want to add one more thing. What is very interesting is that the most influential genetic variants that influence asthma are very close to areas of potential epigenetic marking. In other words, these two things go together. One of the ways in which genetic variation increases the likelihood of being protected or of being at risk is because it alters the potential for certain of these CPGs, as they're called, for epigenetics, to be marked or not marked by environmental factors. So there you have another, what we could call gene by environment interaction.
Push to talk. There we go. So, Fernando, I'm struggling with the final messages from your talk.
Good. That's good.
Yeah, I know. This is the fun part of these processes. So, you are saying to me as a lay public, I'm gonna talk as lay public, that there are individual differences in susceptibility. Is that right?
And in risk and in resilience.
Right. And that actually to push you as a young organism to risk is good within certain circumstances.
Let me put it this way.
Okay. Okay. Because what I'm hearing, before we used to clean everything. And now we're saying, no. Let's not clean it. Let's expose the young organism to these risk factors so they can learn from early on how to deal with them later on in life. And I'm trying to put this on a lay public context and saying, you're really telling me to a certain extent, and I'm all with you, because I remember very early on when I heard that rats who were exposed to stress early on, right, were stronger, and they learn with the environment, stuff like that. So I want to unpackage what you are trying to tell us from this asthma to how do we translate these principles into development, into human development, into what do we do for early organisms.
This was only a 15-minute talk.
I know, but that's why
I
I'm
should have shown
asking
my 1-hour talk, because in my 1-hour talk, I have a study that was done just, I think, 7 or 8 years ago, in which people were made to exercise or not exercise, and then their cholesterol-related, metabolic, a full-blown metabolic assessment was made, and there was a 10% of the population who did worse after they exercised.
Yeah.
But what does that mean? That means that we need to be prepared, because, as you heard very clearly from Phil, if you take the means, obviously the populations as means seem to do better with certain exposures. And culturally, we tend to believe that that is the case. And that is fine. It's perfectly legitimate to have cultural, how could I say, freestanding ethical approaches. The problem is that the population is very varied in its responses to whatever we do.
So should we take not the mean, but the proportion of the population that does better?
In the ideal
and also take and also say, like they do in those very, very rapid advertisements on TV, everything wrong that can go with this great drug for you. Also talk about the ones and try to understand, as you were saying, Phil, who does well and who does.
That is the real thing. How do we do a science that stops thinking about people as the same as the means of the population they live in? That is the essence. But that is very difficult. This is like the old joke of the drunkard searching for the keys where the light was on. Because we study what is easy. You saw what all GWASs are done in urban populations. You go to a rural population, it doesn't apply, because they're exposed to different things.
So as a scientist, I can take that message. I'm trying to figure out, given the mandate of this group, to come up with recommendations. Not for science. For science, we know what the recommendations are. More research, right? And we need to specify what the specific of the most research. We know that. But for recommendations for policy and practice, where does the two positions, the scientists should talk about it too. The two positions leave us. Is it individualized interventions given your specificity of your bio and your environment? And all right,
so
shut up.
I have two things to say. These are great comments. One, I think that one area of what Fernando is talking about, you probably need to hear his 2-hour talk for this. But I think one of the things that's so critical about the research that he was presenting is that it is very clearly understood what the mechanism of early exposure to pathogens is in terms of how it affects the development of the immune system and the immune response.
It's better known.
It's better known. And so I think we're sort of on shaky ground when we say, well, we used to think X, it's like, don't drink wine. No, drink wine. Well, drink this much wine. Those numbers are always changing in terms of how we communicate science. I think when we have a better idea of what the mechanisms are, why is it, first and foremost, that early exposure to pathogens might not be a harmful thing in general, is one message, because we can describe the mechanism in ways that I think are comprehensible to people. And the other is this concept that it's not, I think when we go into differential response, it's not that we don't want to go down the road of everybody therefore needs something completely different, but rather that there are probably going to be a finite number of variations in how people respond to different experiences that allow us to prescribe different sequences of supports at different dosages at different points in development that'll make the biggest impact. And I think if we understand that for some people, certain things might lead to more optimal outcomes than others. For other people, certain things might lead to more negative outcomes than others. But it really helps us to identify that we're not talking. I worry that the personalized approach, once we start getting away from central tendency, is well, you're saying everybody's going to need something different,
no.
and that's going to be impossible, and so you're just one of these people that wants policymakers to devote their entire budget to this stuff. So we do need to become much more precise with this.
Just very briefly, nobody would be alarmed today if by, God forbid, you develop breast cancer, that you're going to be genotyped to treat you. Why shouldn't we think about it the same way with respect to issues that have to do not directly with something that is considered only biological, like breast cancer? I mean, nobody would. I mean, you please go to another doctor if they don't genotype you.
Yes, hi. Going back to the real-world issues, I think we all agree that maltreatment is one of the biggest stressors, one of the most toxic things that a child can experience. And I'm going back to one of your earlier slides, Fernando, where you show these 3 lines going up like that with maltreatment, where basically, regardless of what genotype or genes you have, maltreatment sucks. It hurts. It's bad. Right? And I'm reminded of a report by the Framework Institutes a few years ago on resilience, which said a rising tide lifts all boats. So when you're talking about kids at risk for maltreatment, does it make sense for us to be thinking, well, first let's genotype you and then see whether you're going to be more or less responsive and then decide whether you should get the intervention or which intervention you should get, when we know there are some interventions that do help reduce maltreatment risk among mothers or caregivers. So that's one part of my question. The other part is also about, not just moderators, but mediators, saying, if I know that I can work with Nadine and reduce her risk for maltreatment in terms of how she is with her child, do I really need to understand what's going on in her brain in the real world? As I'm trying to make an intervention that would be disseminated in all of Arizona, is it important for me to make sure that my intervention increases her well-being and reduces the maltreatment to her child? And how much do I really need to look at her genotype, her child's genotype, or with how her brain is firing, or her child's brain is firing?
Answer briefly, because that's for Phil, not for me. But some phenotypes are going to work like height. In other words, with respect to genes. Some phenotypes are going to work with those 3 lines that I just said. In other words, it may be perfectly well that depression with respect to maltreatment works exactly that way, and you don't need to genotype anybody. Some others are going to have a much more complex way of thinking, but I cannot tell you in your area because I don't know. But in other words, in some cases, you won't need to do too much because it's obvious what the result is. In others, it's going to be more complex.
Yeah, so this is the 2-hour talk distilled. I think the question of what it is that translating neuroscience can do for interventions is an extremely important one. We certainly don't want to just add another layer of measurement, especially when it's costly, especially when it potentially could be used as another form of stigmatization for high adversity populations. On the other hand, I think, Sonia, that when you say, if I already know what is needed to reduce maltreatment, that's a big if. And I wonder if the state of the field is really that we do know what's needed consistently, or whether our interventions are as well developed or as precise as we need them to be. I think most of our interventions are very imprecise. They're not particularly efficient. The other thing is, I think oftentimes the attributions that we make, if I can just do a brief example, about what's going on are not necessarily accurate. A good example of this is kids in classrooms with really high adversity maltreatment backgrounds often are described as inattentive, as not engaged, as not caring about or attached to their peers or the teacher. We've collected now data from a number of studies suggesting that what we see in these populations is simply a lack of response when they get feedback from an adult in the classroom. And we see that at a neural level. And what it suggests is that our intervention should be designed to help children separate signal from noise in these contexts, not that they're being disobedient, that we need to focus on figuring out ways to help them become more attached to those in their environment, but simply that it's about being able to tune in more accurately and separate signal from noise. And that's not something that is the path that you go down when you observe phenomenologically what you see in a classroom. It's only something that we discovered as a result of collecting electrophysiological data from these kids.
So how would this be translated in the real world, going to scale?
Right, so then you can have behavioral interventions in the classroom that are very much designed to make feedback much more salient to kids. And so it basically doesn't necessarily lead to biologically based interventions, but it allows you to understand where you need to put your focus in order to have the maximum impact.
Okay, so you have a few minutes left. So we can try to get together.
Yeah, and maybe if folks could keep their questions brief.
Brief, okay. So, Michael, you get 2 words.
Well, thank you both for your wonderful presentation. This question is actually for Dr. Fisher. I think what I heard you said, and I heard Jack Shankov said the same thing, the need for a new kind of research paradigm, a new research approach in early childhood. So instead of trying to find kind of one-size-fits-all intervention, which is kind of what randomized control trials give us. We're trying to figure out kind of what intervention works best for which population under what circumstances, which would then require a more nimble kind of research platform that allow you to continuously adjust, enhance, adapt your intervention in these sort of rapid cycle testing. My quick question is, how do you maintain analytic rigor in such research design? So, for example, what would you do with control groups, right? So, in classic randomized control trial, the reason that you have control group is you want to make sure that everything's held equal except for the intervention itself. But if the intervention keeps changing and adapting and so forth, what would you do with the control group?
No, it's a great question, and I'll answer it as quickly as I can. But just basically to say that this is not at all an argument against the use of existing rigorous methodology, but what it is a really strong call to fill in the space that exists historically between small-scale pilot studies that we typically run that are designed to get enough support that then we can run the large-scale RCT with this vast amount of middle ground where you can begin with underpowered designs that maybe just have pre-post data, but where you're beginning to get some confirmation about the underlying cause and effect between what you're trying to change, whether it's changing or not, and whether that's leading to outcomes. And you then populate as you go further through these cycles, increasing rigor, where you begin randomization, where you're able, as the sample size grows, to continue to add in things like tests of moderators. So it's really about more of a progression of the process. And adaptations are viable. Again, there's an evolving methodology, for instance, adaptive trials that allow you in the context of where you're trying something, if you have a non-response to what it is that you're trying, to then re-randomize individuals to receive variations or supplementation. And I think it's in those contexts. So if it comes across as we're saying RCTs are no longer needed and it's a rejection of rigorous methodology, that's really not what we're intending to communicate, but rather that there's a lot of space that needs to be filled.
We have time for 2 brief questions. So Natalie and then Amy. We're going to be on break.
Great. Thank you for these great presentations. I have a brief question for Dr. Martinez. My question has to do with whether or not the science happening in your lab or other labs are at a stage of being able to inform either the prevention or treatment of racial or ethnic disparities in asthma, since you stated this is a condition with huge disparities in this country.
Yeah, there's work being done by ourselves, but essentially by Esteban Burchard at the University of California, San Francisco, on the applicability of all these concepts to disparities. And it's very interesting because responses to known medicines varies by ethnic background. But that has not only to do with genetics, or not even due to genetics many times, but with another modifying factor, which is social context, exposures, and so forth. So this whole concept of modification by background is one that is really getting into us very strongly. But I want to stress something that Phil said. it's very important to do all this guided by biological background, in my opinion. What is it that is going on that explains what you are seeing? And in that sense, the good thing about the paradigm that I showed is that, as he said, we know rather well now what the mechanisms are of response to bacteria in the environment. So that is what is guiding us for that.
This is probably, we probably don't have enough time to dive into this, but there was one sentence that I highlighted in the paper, Dr. Martinez, that was provided to us as pre-read for today that I would love to learn more about. And that is, it is thus possible that prenatal and early life factors that predispose to the development of metabolic abnormalities may be a common thread for asthma and obesity. The discussion about obesity and the gut microbiome, I think, is very interesting because I think that the whole area of overweight and obesity is one in which we seem to do a lot of blaming and shaming of people, and it seems more complex than that. Is that indeed what you're saying?
Do I have half hour?
Yes, I figured this was not enough time.
Just with a sentence, the most severe asthmatic patients that I see in my clinic are obese, are of lower socioeconomic status, are depressed, are stressed, and don't do well at school. Is that because asthma causes all that? No, because I have a lot of kids with asthma who don't have all those things. We are increasingly biologically finding out, for example, that insulin resistance in early life, before you even develop asthma, is a risk factor for asthma, is a risk factor for obesity, is a risk factor for depression. There are common factors that determine the comorbidity that we're seeing in all these kids. It's not only that one disease causes the other, but it's some common factors that are way up in the causal pathway. If we understand those, we can intervene in ways we can really decrease the prevalence of all these conditions, I think.
Thank you.
So I want to thank the speakers again. It was a great session, and we're on break, Jennifer?
Great. Yes. Thank you, everyone.
Okay.
So we'll take a 15-minute break.
Thank you.
So promptly back here at 11:14 and a half.
11:13.